Yes, Dr. Janet.
Good morning to all. Welcome in today's webinar. I, Junaid Tantre, the co-convener of this event, and I'm delighted to see such a diverse and engaged audience joining us today. Your participation enriches our conversation and underscores our common commitment to advancement in our fields. I anticipate productive and collaborative exchange as we begin this learning experience together. SPSR is recognized for its captivating webinars, solidifying its position as a premier platform for professional development in the pharmaceutical industry. I'm confident that today's webinar will continue to reflect our high standards, offering its significant insights from an esteemed speaker. Let us seize this opportunity to acquire new information, engage meaningful, and advance our field. Welcome, and I anticipate a rewarding session for all attendees. Thank you for being here. Today marks an important occasion as we convene for the 57th SPSR International Webinar on Dendrimer Technology Concept to Cutting Edge Applications. This webinar is a part of prestigious series organized by Society of Pharmaceutical Sciences and Research with Dr. Shashi Alok sir, the National President of SPSR, providing respected mentorship. With his leadership, SPSR remains instrumental in promoting progress and creativity in pharmaceutical sciences. And the SPSR is committed to the progression and enhancement of pharmaceutical field through diverse strategies. We aim to facilitate conferences that promote pharmacy-related initiatives and endorse programs and activities addressing public health issues in India, with their emphasis on disease prevention. We also make it a point to acknowledge and celebrate pharmacists whose significant contributions inspire further advancement and spread of pharmaceutical knowledge, ensuring a consistent and rich information regarding pharmaceutical practices and services. It's a truly great privilege to introduce Dr. Mayank Kumar Singh, sir, Principal Scientist, R&D, the National Dendrimer and Nanotechnology Center, Michigan, USA, as a distinguished speaker. Dr. Mayank, sir, expertise greatly contributes to our event, providing substantial knowledge and insights. His participation is certain to enrich our discussions and expand exploration of dendrimer technology. We are grateful for his engagement and anticipate an enlightening session. We are honored to welcome Mrs. Monika Sabharwal, ma'am, as the convener of this webinar. National Secretary SPSR and Managing Director of IJPSR and IJP. Her extensive contributions and leadership reflect her dedication to pharmaceutical sciences and research. We are fortunate to have her expertise steering our session today. I'm pleased to invite Mrs. Monika Sabharwal, ma'am, to warmly welcome our distinguished guest speaker, Dr. Mayank Kumar Singh, sir. Ma'am, over to you. Monica, ma'am, you have to go.
Thank you, Mr. Junaid Tantre. Hello, everyone. I am Monika Sabharwal, the convener of 57th SPSR International Webinar. On the behalf of, I'm delighted. Pardon? Pardon, Mr. Junaid Tantre.
Please continue, ma'am.
Okay. On the behalf of Society of Pharmaceutical Sciences and Research SPSR, I am delighted to welcome you all on today's session titled Dendrimer Technology Concept to the Cut-Edge Applications. We are privileged to welcome Dr. Mayank Singh as one of Principal Scientist, Research and Development at the National Dendrimer and Nanotechnology Center located at Mount Pleasant, Michigan, United States of America. Dr. Singh brings a wealth of knowledge and experience in the realm of dendrimer technology. His innovative contributions to this area make him an incredible valuable source of information, offering deep understanding into their revolution uses to the diverse industry. Today, we will explore the fascinating world of Dendrimer technology, understanding its fundamental concepts, and delving into the cut edge applications that are revolutionizing in the field of nanotechnology and beyond that. Dr. Singh will guide us through this intricate subject. Diamonds will becoming pivotal in the various factors, including drug discovery, gene therapy, diagnostics, and even material sciences. Dr. Singh will elaborate how these nanostructures are engineered and their biocompatibility and their multifunctional capabilities, which make them suitable for targeted and efficient therapeutic interventions. Moreover, he will discuss the latest advancements in the Dendrimer technology, showcasing real world applications and breakthrough research that are setting new benchmarks in the medical and the pharmaceutical sciences. The session promises to shed light on how dendrimers are poised to address some of the most challenging issues in the healthcare, offering a promising solutions that were once thought to be impossible. We are thrilled to have received the 2000 registration. It is a testament to the global interest and the significance of this topic. This milestone, our 57th SPSR International Webinar, marks another significant achievement in our journey. The overwhelming support and the feedback from the global audience continue to inspire and motivate us. The SPSR has been dedicated to advancing the pharmacy profession, and efforts have been reflected in the tremendous support we receive for our webinars. Your continuous engagement and the regular attendance at our sessions, even on Sundays, are deeply appreciated. This dedication from our audience creates a positive impact and encourages us to strive for the excellence in every event we organize. We are greatly honored to invite Dr. Shashi Alok, the esteemed National President of SPSR. His insightful leadership has played a pivotal role in effecting an extraordinary metamorphosis at the SPSR. Established, it is a paragon of excellence in the pharmaceutical sciences. I would like to extend my heartfelt thanks for significant contributions made by Mr. Junaid Tantre, the co-convener of this webinar, along with Dr. Pradeep Gulani, Mr. Ankur Agarwal, and all SPSR team members. Their relentless dedication has been instrumental in bringing the vision of this event into active accomplishment. The seamless execution of this webinar is a testament to outstanding efforts of our team working behind the scenes. Their dedication to the high quality work and persistent effort have been pivotal to make success of this event. My gratitude goes to each member of SPSR team for their careful management of the details and enduring enthusiasm that brought this webinar to future on. Stay ahead in the field of innovation with the SPSR. Join our YouTube channel for the unique webinars, expert perspectives, and the recent findings in the pharmaceutical sciences and healthcare. Engage with a vibrant community that is involved in the conversation driving the healthcare progress. Keep up to date by subscribing to our YouTube channel and turning on the notification bell icon for immediate alerts. Follow our progress in advancing the pharmaceutical sciences that will benefit the society overall. Without further ado, I would like to extend the warm welcome to Dr. Mayank Singh. We are eager to hear from you and learn about the groundbreaking advancements in the dendrimer technology. Thank you for joining us today, for your continuous support at the SPSR's initiatives. Over to you, Mr. Junaid Tantre.
Thank you, and I appreciate Mrs. Monika Sabharwal, ma'am, granting me the opportunity to present this webinar. And now we will start with a short introduction to guest profile. Dr. Mayank Singh, sir, is a distinguished principal scientist and National Dendrimer and Nanotechnology Center in Mount Pleasant, Michigan, USA, with a joint appointment as a graduate faculty member at Central Michigan University College of Medicine and a consultant role at the World Health Organization in Geneva, Switzerland. Dr. Singh's multifaceted career exemplifies his commitment to advancing scientific research and global health. Holding an extensive background in nanomedicine, Dr. Singh's expertise spans both academic and corporate sectors. He's a pharmacist by training with a PhD in pharmaceutical sciences and nanotechnology and holds certifications in intellectual property rights and patent law. His innovation work in dendrimer technology and nanomedicine has resulted in multiple patents and the development of commercial products. Dr. Singh's contributions to the scientific community are further demonstrated through his role as an expert faculty member in chemical biology and biomimetic chemistry at Faculty Opinions Limited in UK, where he performs post-publication peer reviews. His affiliations with numerous scientific organizations and government bodies across the UK, USA, Europe, Japan, Canada, Singapore, Switzerland, and India reflect his global influence and commitment to scientific excellence. In addition to his research endeavors, Dr. Singh has been associated with the Drugs Control Administration, Government of Telangana, and the Central Drug Standard Control Organization, Hyderabad. His professional recognition include being a registered scientist, chartered scientist, pharmaceutical science scientist, fellow of Indian Chemical Society, and an honorary biographical listee for outstanding achievement in nanomedicine. Dr. Singh's passion for scientific innovation is matched by his dedication to education and mentorship. Certified as an academy supervisor and mentor by Publion's Web of Science and a certified peer review for major scientific publishers such as ACS, RCS, Wiley, AAAS, Springer Nature, MDPI, Taylor Francis. He actively mentors MS, MD, and PhD students whose research on brain targeting holds promise to the novel treatment for brain tumor. In his capacity as a mentor, Dr. Singh has guided numerous students and young researchers, fostering a new generation of scientists dedicated to exploring the frontiers of nanomedicine. His numerous accolades and memberships in prestigious scientific bodies underscores his contribution to the field. Dr. Singh is an active member of several professional organizations, including American Association of Advancement of Science, the Royal Society of Chemistry, and American Chemical Society. His work has been widely published in top-tier scientific journals, and he frequently presents at international conferences, sharing his insights and advancing the discourse in nanomedicine and pharmaceutical sciences. Uh, we are pleased and proud to announce that over 2,000 attendees have registered from around the world, representing a wide variety of nations. Notably, we have substantial participations from the countries like India, Egypt, Indonesia, Iraq, Kuwait, Libya, Malaysia, Nepal, Nigeria, North Macedonia, Pakistan, Saudi Arabia, Syria, Tanzania, UAE, USA, and Vietnam. We are profoundly touched by the significant and diverse reaction we have received. Our sincere thanks go out to every individual who took the time to engage in this important occasion. Thank you for all joining us today. Uh, let's make this webinar an insightful and enriching experience by everyone involved. We have our esteemed keynote speaker today, Dr. Mayank Kumar Singh, sir, with us. Welcome, sir, to SPSR. Your insights would be highly valuable, and I would appreciate if you could present them in speech. Over to you, sir. You can share your PPT, sir, presentation.
Thank you very much, SPSR team, Junaid ji, Monika ma'am, and Dr. Sasi Alok sir. And thanks for a wonderful invitation on this occasion of Guru Purnima Day. Really, just 1 hour ago, I came to know that today is Guru Purnima Day, and then I decided to share my. Can you see my slides?
Yes, of course, sir. It's visible. You can put it on
It's visible.
display.
Is it
Yes,
moving?
yes, sir. It's
Okay.
moving. Absolutely.
Yeah. So thanks for all this invitation and warm introductions. So as being today the Guru Purnima Day, I decided to dedicate my this talk to Professor Donald H. Tumalia on his 18th birthday for his outstanding achievement in advancing the field of dendrimer. And he is known as the father of dendrimer technology. So today I will be delivering my talk dedicating to his 85th birthday on the dendrimer technology concept to cutting edge applications. So my talk will be divided into two sections. The first section will be the scientific journey of Professor Donald Tumalia, who is known as the father of dendrimer technology. And the second section will be my scientific journey with dendrimer from India to USA. So currently we are located in Michigan, United States, and were established in 2002 by Army Research Laboratory in association with US FDA and NCL. And we have our funding agencies like Defense Advanced Research Projects Agencies, National Cancer Institutes, and National Institute of Health. And I would like to introduce about the early research into dendrimer technology. In 1979, Donald Tomalia and James Dewald make a breakthrough by adding a methanol as a solvent to normal polymer production procedures in Dow Chemical Laboratory that created a discrete units in which they had two branches. The right side figure, you can see a branch structure and a computer simulation figures that when they added a methanol in a regular polymer productions, they found something kind of a discrete flexibility in the polymer structures. And from there, they got original idea how to make a dendritic patterns. This is the original peak of Dow Chemical Laboratory of 1979, the group working on dendrimer technology. And this was the birthplace of the dendrimer in Dow Chemical Company, which is located in Midland, Michigan, USA. And it is almost 15 miles, 15 to 20 miles near from Mount Pleasant. And in 1984, Donald Tomalia published his first paper by saying that dendrimers are the fourth, by classifying dendrimers as a fourth class of polymers, which comes as synthetic macromolecule after linear, cross-linked, and branch structures. You can see the middle structure and see how the progress in polymers have been done from the linear, cross-linked, branched, dendritic polymer, and finally the dendrimers. And the word dendrimer coined by Tumalia et al. In 1984 originated from two words of Greek words. Mainly dendri means branched and word means meaning of, part of. And this appeared in the American Heritage Dictionary and Oxford Science Dictionary in 2005. And this word dendrimer was accepted from 2005 all over the world. And this is one video which represents the dendritic structure and its scope in drug delivery and other purposes. And so in 1985, around developing of dendrimers, Tomalia divided this dendrimer synthesis in two structures. One way, Don Tomalia was exploring this technology in the field of dendrimer by saying asymmetrical branch cells, while other groups we started working on this concept in case of saying asymmetrical branch cells. But the major advantage of the symmetric branch cells was that they were having the cavity, and almost these were the dendrimers which reached to the commercial stage and come in the market. And rest of the dendrimers were not having the cavity to load or release the drug. So today our whole lecture, whole talk will be basically on the symmetric branch cells dendrimers, which is known as polyamidoamine dendrimers. And later in 1993-94, this dendrimer started commercializing with Sigma Eldridge, and it started going on increasing through all over the worldwide. And then it started exploring other applications and areas in case of personal households, energy, electronic, food, agriculture, chemicals and manufacturing, environment, medical and health. So it was a multifunctional application of dendrimer started initiating due to the commercialization of dendrimers. This is the architectural view of the PMM dendrimer. A PMM dendrimer is mainly consist of a core, anterior void, and a terminal groove. And this is mainly a synthetic nanosized molecule which has capability to entrap a molecule inside in their interior voids. And also we can surface engineer the other molecule by chemical conjugation on the surface. In a common way, we can say this is exact like our laddu, where sugars are embedded in that. And in another way, 3D simulation, it is just like a buckyball where we can see how a drug can be entrapped in a buckyball. This is the physical association concept of dendrimers. how host-guest binding takes place, and physical encapsulation happens. A hydrophobic guest, this is one of the best host-guest interactions where we can say how the encapsulation of drug, entrapment of a drug takes place with dendrimer. And then we will come to the synthesis of dendrimers. The synthesis of dendrimers are mainly done in the divergent, convergent, and double exponential synthesis. Divergent means it starts from the core and goes to the surface, while the convergent is from surface to the core. And while the double exponential is the combination of divergent and synthesis. And this is the synthesis of dendrimer and their molecular weight. We will start from the core and start reacting with Michael addition amino group with methyl acrylate and keep on adding one group, washing it, second group, third group, fourth group. And we can see as the generations, as the surface groups increases, the molecular weight increases. From the table, you can really see how the generation 0 have a 4 surface group, molecular weight is this, and on generation 1, 2, 3, it keeps starts increasing 8, 16, 32, 64 functional group, and the molecular weight start enhancing. So this dendrimer synthesis is just mainly based on the concept of mathematically. You can design dendrimers the way you want. And here you can say how the particle size of the dendrimers increase from G0 to G8. You can see how slowly the size increases from G1, G2, G3, G4 with 4 nanometer, 5 nanometer, 6.6 nanometer, and up to 10 nanometer. We can synthesize dendrimers from 0 to 10 nanometer with 10 generations. And then we can further functionalize the outer surface of the dendrimer with amine, carboxylate, neutral pH like tris, amidoethanol, and pyridone surfaces. And then we can further characterize with TEM, AFM to confirm their size are within 5 to 10 nanometer depending upon the generations. Then we can see a comparative distribution of size exclusion chromatography where a traditional linear polymer is polydispersed, whereas the dendrimers are the monodispersed, and we can see how small and how precise their patterns are as compared to the traditional polymers. And then they can be further identified, further characterized with ESI electron space ionization concept. This is the multi-top analysis of PMM dendrimer, where we can say how the experimental and theoretical value has been calculated, and almost 90% of the dendrimers have been found to be successful in the synthesis. And then it was the HPLC analysis of G5 PMM dendrimers, where we can see how the growth in the area under curve had been seen as compared to the different generations. Now we will talk about the PMM dendrimer as a multifunctional platform. As we regularly use our mobile phones, and we can easily say that we can do a multitasking work with our platform, with our mobile as a multifunctional platform. In such a way, we are here to explore the dendrimer technology as a multifunctional platform where targeting, imaging, therapy, diagnostic can be done with one single entity, that is dendrimers. This is one of the intravenous injections of hydroxyl dendrimers from Aswatha Therapeutics. And we can say how the dendrimer-based technologies travels in the body and distribute to the specific target. It starts circulating all over the body, blood vessels. You can easily see the dendrimer is traveling inside the blood vessels. Hydroxyl dendrimer with payload attached on the surface. This is a different multifunctional platform technology where the drugs will be attached on the surface of the dendrimer. You can see the drug and the dendrimer. It is a chemically conjugated concept. Now it will travel healthy cells and inflamed cells in case of cancer and all. And we can see how it goes under lysosomal pathway. Very beautiful 3D demonstration will clearly show how dendrimer reaches the lysosomal pathway, follows the lysosomal pathway and enters in the cells. These dendrimers are almost neutral, not having any toxicity and clearly excreting through the urinary tract. It can cross the blood-brain barrier also. Target sites of inflammation in the case of cancer and across the blood-brain barrier. It can use to cross for the blood-brain retinal barrier also. Arthritis. And now what we discussed in the video, it is the same here. PAMM dendrimer as a multifunctional platform for drug delivery. You can say how a folic acid has been coupled with acetomido and the fluorescein, means targeting drug delivery and tracking can be done with one delivery system at a same time. So in a 3D diagram, we can say how the drug can be loaded physically and it will follow the hydrogen bonding or ionic interactions or molecular encapsulations. Whereas in another way, we can chemically conjugate the drug and we can see the therapeutic effect. And what will the mechanism of drug release, a pH dependent drug release? We can say at neutral pH, the dendrimers will be in normal and the backfolding happens and start releasing in acidic environment. So most of our cancer environment is acidic in pH. Even if we make a oral drug delivery system also, it will go in our stomach and from there go to intestine and it will release that drug. So this is one of the pH technology, the pH dependent based drug release mechanism for releasing the drug to the specific site. And this is the periodic property of PMM dendrimers from G0 to G10. So we should always have a criteria that how which generation of dendrimers can be used. You can see as we are increasing from G0 to G10, the size increasing. Being the size increasing, the dense packing, the structures, the surface of the dendrimers become so rigid and they don't allow to enter the molecule. So you can see generation 4, 5, 6. After generation 5, 6, the size start increasing and the surface area are getting so much compact. So static hindrance and lot of things happen. So depending upon our utilization and depending upon our purpose, we have to choose a specific size of dendrimer for specific delivery. And this is the payment dendrimer as a artificial proteins. This was the first example of synthetic monodisperse polymers that closely mimic the biological proteins like hemoglobin and hematocytin, and we can say many things, DNA, immunoglobulin, insulin. So this is the beauty, this is the main feature of dendrimer which mimic the biological proteins. And this was the first example of dendrimer modified monoclonal antibody with polyvalent surface of the dendrimers, and on this base few products, few technology have been came in the market, which we will see in the coming slides. And this is the primum dendrimer based MRI images where different generations of dendrimers have been used, and where you can say by increasing the generations there is high contrast in case of clarity of the visualization of the images. So this was the one of the main feature of the dendrimer for as using as a MRI contrast agent. It enhanced the a contrast of imaging of the MRI technology. Further, this technology was also used to identify the conformation of the um, lymphoma in lymphonides, with the lymphoma based animal lymphoma models, where we can say the arrow clearly shows the swelling of lymph node. In another way, a cisplatin induced acute tubular necrosis was also confirmed using the G4 dendrimer contrast MRI technology. Further, in 2D of micro MRI of liver metastasis also confirmed the high contrast imaging with this dendrimer technology. And then what was the vision? Don Tomalia at that time thought that after developing a dendrimer, it goes to the in vitro, preclinical, clinical, FDA approvals. And this was his vision that around 2020, after 20, between 210 to 220, the product should start coming in the market if everything goes well. And to be very accurate and to be very perfect, that dendrimer technology based status CS acute cardiac diagnostic system was launched in the market, and it was mainly one of the troponin analyzer in case of the heart attacks. It takes mostly 5 to 7 minutes to confirm the status of the cardiac arrest in the patients. So, and this is the few upcoming dendrimer-based cosmetic products with Unilever, Johnson Johnson, Revlon, and L'Oreal. These are few are in clinical trials, and this is Viral Easy, which was launched in 36 countries during COVID-19 as a preventive anti-nasal spray. And Vivazell has almost cleared its clinical trial phase 3, and now it is in the stage of coming in the market. And there are some Vivagel based condoms, and there are some anti-cancer drug deliveries, which is on the way. And this is Professor Tumalia, and where we celebrated his 85th birthday in the center. And now I will start my background. I am basically from Mirzapur district of Uttar Pradesh. I completed my schooling from CBSE board in Northern Coalfield Limited premises. Then I got my pharmacy training in Uttar Pradesh in Bundelkhand University, Jhansi, and then I moved to Andhra Pradesh, Hyderabad for my PhD program, where I was associated with Drug Controller Administration Telangana and CDSO Hyderabad. And I further completed my certification in patents law with National Institute of Hyderabad. And this was my scientific journey. I would like to showcase my scientific journey with Pamem Dendrimer for master dissertation during 2010 and 12. I remember during my master's entry, Professor S.K. Prajapati sir asked me to read these few articles, and he gave me these articles. And you don't believe these articles changed my complete scenario, what to do in future, how to do, what to do. Still, I got this in my file, and I thought to showcase these articles from where I started my dendrimer journey. And you can see the first article where Dr. Tumalia is clearly saying about the birth of a new macromolecules, and where in the next article he described about the dendritic state. And third was the Sigma Aldrich article, where Dr. Tumalia and his team with Professor Chauhan and Lori Raina and all describing and exploring the dendrimer technology. And then I started networking during my master dissertation, attending the conferences and start meeting the distinguished professor and scientist of India. You can see in the first top, Professor Chauhan meeting me with Dr. N.K. Jain, Dr. Tripathi. And here I started my scientific journey and started understanding how science move and what are the things required and how mentors are very important for you. And then I got my first paper published. I got only one paper published, I will say, in my master's and one paper in my PhD. And this master's research was done at Bundelkhand University, and my PhD research was done at CSIR IICT Hyderabad. And then during my PhD, I traveled for showcasing my uh, PhD work in International Dendrimer Symposium, where I met Professor Donald Tomalia. I showcased my work, I gave my talk, and he was very much impressed with my work and my concept, and he offered me to join his organization after my PhD. And after completion of my PhD, I reached to the birthplace of PM Dendrimers with father of Dendrimers. You can see it took me 1.5 years to reach there after my PhD, and finally I started working with Professor Tumalia, and then we start exploring dendrimer technology for COVID-19, and then later we made the I can say the first image where you can say this was the third article which was given by Professor Chapam Prajapati at that time to read, and then after coming here we developed a hybrid dendrimer technology, supramolecular assembly, and these were published by Sigma Aldrich. And now I will try to showcase one of my recent work on resveratrol with permanent dendrimers, uh, where you can see that resveratrol is mainly found in the blueberries and the grapes, and it has a very less water solubility, low permeability, and stability issues. So we tried to use dendrimer multifunctional technology for delivering it to the topically in the form of cream and gel, and we were able to enhance its solubility by 3000 folds, increase its resolution, topical permeation, and sustainable, eco-friendly, and biocompatible. And where you can see the first the solubility profile, almost resveratrol is insoluble, and by using the dendrimer resveratrol dendrimer technology, we were able to increase its solubility by 3000 fold, enhance its dissolution almost in 20 minutes. Dendrimer resveratrol complex was able to dissolve in water as compared to the pure resveratrol. And then we try to improve the solubility, enhance the permeability, and see the pharmacokinetic profile, how it alters by using the resveratrol dendrimer technology. And I really thanks Professor Chauhan for involving me in this anti-aging project since 2013, and it took 10 years to bring this product in the market. Professor Chauhan started this work in 2013, and I was his PhD student at that time, and now this product is in the market, recently launched with Amazon in US. And further then again, the few case studies were done with this technology, and then we published this article recently in JDST, and I really thanks SPSR team for giving me opportunity to deliver my talk to this platform. Thanks Professor Chauhan for his mentorship. Thanks Dr. Diwan for guiding me, supporting me, and thorough mentorship. I thanks Bundelkhand University staffs, ACSIR, IICT, Institute of Science Bangalore, Aswatha Therapeutics for showcasing the therapeutic potential of hydroxyl dendrimers, Siemens, Star Pharma, Central Michigan University College of Medicine, Nanosynthans, World Health Organizations, and I thanks to all the pharmacy department of Bundelkhand University, and Dr. Sasi Alok sir might be here, Dr. Prajapati sir is here, and today is the Guru Purnima, and it was really a great day to bow my head to all my gurus and get the blessing from this. And thank you very much.
Thank you so much, sir. It was nothing sort of exceptional. The journey you shared might inspire others or us also to to look for your passion and fight and get to know what we are interested in. Thank you for sharing this, sir. It was interesting one. So at the end, we have collected some of the questions, sir, and I'll be asking you some questions. So one of the questions we have received is, how can dendrimers be functionalized for specific applications in medicine and biotechnology?
Yes, this is very, very good questions, as I had shown the video, right, where you had seen the dendrimer functionalizations. So if we are targeting some kind of diseases, if we take the example of cancer, right? So in case of cancer, there are many receptors which are overexpressed. For example, folic acid, transferrin, and other things. So we have to couple that targeting ligands which is specific to that receptors. So this is one of the things, coupling of the targeting ligands with the dendrimer surface, and then you can that dendrimer based targeted system will go specific to the specific disease or specific site.
Sir, one more question is there that how do dendrimers enhance the delivery and efficacy of drug? No, in a short word.
So this is one of the best question I will say, because dendrimer is totally different from the other delivery system. For example, if I will talk about the liposome, solid lipid nanoparticles, it is just like if in a layman language, I will say it is like a samosa, where your stuff is inside some cavity, right? And how much you just put it inside, you think it goes, it has a leakage issue and all those things. But dendrimers are not there. The dendrimer surface is, it have a primary amine, tertiary amine, and other surfaces, surface groups. So the amine groups helps to enhance the solubility, and other surfaces group, primary amine, tertiary amines, and other groups helps to make a physical or hydrogen bonding kind of things and encapsulate in the system. So it has a double kind of interest, you can say. It will entrap also, and it will enhance the solubility also. So this is one of the mechanism of dendrimer which is not possible with other systems.
Right, sir. Great. Sir, one of the question is, can you share a case study of successful application of dendrimer technology in healthcare? Although you have shared in
Yeah, I had shared
PPT
my
also.
recent work with resveratrol topical, a case study, so it is already there. And still there are many ongoing, like Star Pharma's docetaxel drug delivery system. Lots of case studies are on the way in the
Right,
area
sir.
of cancer technology.
So one more question is, beyond medicine, what are some emerging industrial applications of dendrimer technology?
Okay, beyond medicines, dendrimers are used in semiconductors. Dendrimer are used in inkjet, in many places, many places. Oh, I had shown you the slide, like home case applications, in spray, in dryer, and in it has a lot of applications. It has an anti-coating in polymer industry, paint industry, in ceramic industry, in diagnostics. Drug delivery is already known.
Right, sir. Thank you once again, sir. As we draw our 57th SPSR International Webinar to a close, I would like to extend a heartfelt thanks to all our distinguished speakers, participants, and organizers who have made this event a resounding success. Today's webinar on dendrimer technology concept of cutting-edge application has been a remarkable journey through the fascinating world of dendrimers, highlighting their incredible potential and versatility. Our discussions have underscored the transformative impact of dendrimer technology across various domains from drug delivery and diagnostics to nanomedicine and biotechnology. The insights shared by our esteemed speaker have illuminated the fundamental principles behind dendrimer technology and showcased cutting-edge applications that are at the forefront of scientific research. We have explored the intricate design and synthesis of dendrimers, their functionalization for specific applications, and their remarkable ability to interact with biological systems at the molecular level. This webinar also highlights the collaborative nature of scientific advancement. The interdisciplinary approach and international collaborations presented here today are testament to our collective effort required to push the boundaries of what is possible in dendrimer technology. So as we conclude, let's carry forward the momentum generated by today's discussions. Let's continue to explore our new frontiers, foster collaborations, and translate these scientific advancements into practical solutions. Thank you to our esteemed speaker once again, sir, participants also for their valuable insights and contributions. We're deeply thankful to Dr. Shashi Alok, sir, the National President SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance as we offer profound thanks to our scientific team. We are truly thankful for the contribution of our keynote speaker, Dr. Mayank Kumar Singh, sir, whose expertise provided us with a valuable understanding of dendrimer technology, concept of cutting-edge applications. We are grateful for the significant involvement in the 57th SPSR webinar. We're also grateful to Mrs. Monika Sabharwal, ma'am, also for giving their expertise. So thank you once again, sir. It was really, for me personally, I have been more exposed to dendrimers and I'm very fascinated. And particularly your journey, sir, it will definitely inspire many others to follow the passion, sir. Thank you once again, sir.
Thank you very much, Junaid ji and team and SPSR team. I really thanks everyone, Dr. Sasi Alok sir, Monika ma'am, you, and complete entire team and audience for hearing me and giving me opportunity to showcase my scientific journey here. And once again, thank you very much. Have a great day. Yeah.
Thank you so much, sir.
Yeah.